So there are various number of cloning enzymes. So when it comes to the definition of the cloning enzymes are a group of enzymes used in molecular biology to facilitate the cloning and manipulation of DNA. So the various cloning enzymes are nuclease, polymerase, ligase, transferase, kinase, and DNase, RNase, and phosphatase. Uh, when it comes to nucleases are the enzymes used to cut phosphodiester bonds in DNA molecules. Function of nuclease is non-specific. Exonucleases. There are exonucleases. Exonucleases are the enzymes that degrade the DNA or RNA molecules by removing nucleotides from the ends. So these are the examples of nucleases. Endonucleases. As the name suggests, endonucleases cleave DNA or RNA at internal position rather than the ends. To cut single-stranded RNA, S1 nuclease and mung bean nuclease are used. They can be utilized to remove unwanted single-stranded regions to create blunt ends in DNA fragments. S1 cuts at 3' end while mung bean at 5' end. To cut double-stranded DNA, DNAs which are specific and restriction endonucleases are non-specific are used. Polymerase form phosphodiester bonds in DNA molecules to extend the chain of DNA. DNA polymerase catalyzes the synthesis of DNA strands using template DNA strand and nucleotide triphosphates. In cloning, DNA polymerases are employed in several applications such as PCR and DNA sequencing. Tag polymerase. Tag polymerase, derived from the thermophilic bacterium Thermus aquaticus, is one of the most widely used DNA polymerases in PCR. It exhibits robust activity at high temperatures, making it suitable for DNA amplification in thermal cycling. RNA polymerase. RNA polymerases are enzymes responsible for synthesizing RNA strands using a DNA template in cloning. RNA polymerases such as T7 RNA polymerase are used to produce RNA molecules from cloned DNA templates. T7 polymerase is for replicating viral genome. Ligase. Ligases help in formation of phosphodiester bonds between two molecules. The fragments extracted may have different ends, that is blunt or cohesive ends. So this is the difference between two types of ligases, DNA ligase and T4 DNA ligase. DNA ligase isolated from E. coli, whereas T4 DNA ligase is isolated from T4 bacteria. It can form 5-phosphodiester bond between cohesive fragments. It can form phosphodiester bonds between both cohesive and blunt fragments. Ligation efficiency is better than T4 DNA. Ligation efficiency is poor when compared to DNA ligase. Transferase. Transferases are used to add nucleotide towards the 3' end. The type of transferase used for gene cloning is called terminal transferase. So this is the difference between DNAs and RNAs. DNAs cut phosphodiester bonds in DNA. RNAs cut phosphodiester bond in RNA. Target more towards double-stranded DNA. Target more towards single-stranded RNA and less towards double-stranded DNA or RNA. Types of DNAs. DNase 1, DNase 2, mNAs. Types of RNAs are RNase A, RNase C, RNase H. So these are the differences between kinases and phosphatases. Kinases adds PO4 group at 5' end of DNA molecules, and phosphatases removes PO4 group at 5' end of DNA molecules. PNK means polynucleotide kinase can add PO4 to any DNA molecule at 5' end. The process of removal of PO4 at 5' end is called phosphorylation.